So this asymmetric PCR is another kind of PCR. Why we call it asymmetric? Because one of the strands we amplify more than the other strand. How we do this? The primers we add, the strand we want more are the abundant primers, okay, and the limiting primers are less. So usually we do this when we, for example, sequence. In sequencing, we only have to use one strand. We have to amplify one strand. Here we use an asymmetric PCR, only one strand. Okay, or as I told you, if you want to clone a gene on 5' and 3' gene is degrading on both sides, the mRNA is degrading, so we can use asymmetric PCR to amplify it. Or if you want to incorporate a mutation, so what you do, you amplify half of the gene first, then the other half, then use a ligating primer with the mutation, and this ligating primer then have incorporated, and the new mutated sequence will be here, and then the gene that we have will have the desired mutation. So then another kind of PCR is your long PCR. So why we call it long? Because the PCR has processing, the Taq polymerase has processability, and after say adding say 700, 800 base pairs, then the efficiency of the Taq polymerase will fall off from the strands. So for these type of reactions, or genes are very long, what we do, we use EFU polymerase or VNTR polymerase. These are polymerases that have high efficacy. Okay, and they are also able to synthesize longer DNA strands. So if you have a DNA strand that's over say 10 kb, you'll be able to amplify it. However, if you will do the same with the Taq polymerase, the normal Taq polymerase that we have in the lab, you will not be able to do it because with time the efficiency and the efficacy of these polymerases decrease. Okay, let's do this. Then you have the anchored PCR. The anchored PCR, if you remember when we started cDNA synthesis, when we were cloning the gene of interest that can be had from what we just gene cloning, you have one strand and which one of the sequences known at one end. Like for example, you have an mRNA where you know that at the 3' end you have poly A tail, right? Then the primers, and you know the prerequisites of PCR are that you need to have a template strand and you need to have a free 3'OH group that is provided by the primer. So the primer is here, and from 5' to 3' it will you have a poly A tail here, right? And now you can use oligo-DT. You can also use oligo-DT here, primer, oligo-DT primer. Right? So this is the 5', and now you have a free OH group. Okay. This OH group now will provide 3' OH. Okay, this on 3' OH of the ribose. To this, your DNTPs will bind and you'll synthesize the second strand. Okay, to this they will bind and they will form the second strand. Now, once the second strand is synthesized, you want to amplify other strand because you know you want to have forward and reverse primers to amplify, to completely amplify your DNA fragment. If you only have one side primer, then you will be an asymmetric PCR. So now what you do, you add Gs, okay, DGTPs to this mixture and thermal transferase, and that will add GGs here. Okay, then you can add a primer like CCC to amplify this strand. So these type of PCR are called as anchored PCR. Now, another type of PCR is your allele-specific PCR, in which, for example, you have two alleles or alternative forms of a gene, and these alternative forms of a gene can be due to mutation. Okay, when you have a DNA strand, you have a G here and you have a C here, right? You use 4 sets of primers here. Okay, so if you amplify with primer P1 and P2, what will happen? All the DNA that's between will be amplified, and you have a larger band that you will have here. Okay, this band will be non-specific because it's not, the primers are not falling at the allele, right? Now, if you use P3, okay, P3 and P2, again, this will have an A here. Okay, if this has a T here, it will have an A here. So this will be able to amplify this primer. Similarly, if you have a C here, okay, in this primer, it will not be able to anneal here because you have a T here. To anneal, it should have a G here, right? G will pair with C. But when you have a C here and a T here, it will not be able to anneal, right? So this band, this amplification with this and this will not occur, and this will not occur because forward primer is there, but reverse primer is not amplifying because it's not ligating, it's not annealing. However, these two fragments will be able to form the DNA. In this case, if you see, you have band from P1 and P2, that's here, okay, and P1 and P4, that's here. Okay, this is your GG homozygote. Now, if you use the same P4 primer here, you have a band from P1, P2, that will be here. You have a band from P3 and P2, that will be here, and then you will have a band from P3 and P2, okay? 1, 2. Okay, you have only 2 bands in this case, right? So again, you have 2 bands here. If you have a heterozygote, okay, you have an amplicon from P1 and P4, P1 and P2. Okay, these are the 2 bands. Okay, then if this is ligating to one of the strands, you have an A here, you have a T here, then it will again form a band. Okay, so then you will have 3 bands if it's a heterozygote. If you have a homozygote, you have these 2 bands, and if you have a homozygote, you have these 2 bands. Then other type of PCR is your tie-down PCR. Tie-down PCR usually is used for increasing the efficacy of polymerase because these primers that use in the PCR reaction, they have a TM. TM is a temperature at which these primers tend to bind to the DNA. Okay, the TM as such can be defined as temperature for melting, and this temperature for melting is half of the temperature at which half of the DNA strand is denatured. Okay, so when we get a primer set, it's written that the TM is that. So we use usually 2 degrees below that TM. Okay, that will be the annealing temperature for these primers. So in tie-down PCR, what we do is if we run a PCR reaction at 63 initially, with each cycle, we will go down the temperature. The temperature will decrease the temperature because once we have a lot of strands of these amplified regions after say 5 cycles or 2 cycles, then another 25, we run 30 cycles or 35 cycles. In the first 2 cycles, we keep the TM say 63 to 59. But in next 25 cycles, we keep the TM at 58. Okay. So this is called as a tie-down PCR. This increases the efficacy of your tag polymerase. So VNTR PCR, VNTR stands for variable number of tandem repeats because, for example, you have these are repeats like you have ATTCG, ATTCG, ATTCG. So how many times these ATTCGs are present in a particular organisms? The length of these we use to determine. We can use these as markers also. Okay. So what we do, we use primers because the regions that are flanking these VNTR regions are conserved. We use primers against these forward and reverse primer. And when we amplify these, as many, if these are these ATGCs are many, say 50, 100, 200, then you have a larger band. If you, or a larger band. Then if they are small, they are less numbers, say 50 or 10 or 5, then you have a smaller band. So depending on the length of these tandem repeats, you'll be able to determine different organisms that which strain they belong. Again, assembly PCR is the PCR that we use to join two different fragments. Okay, if you have a larger fragment to clone, it's very difficult, or a larger fragment to amplify. So what you do, you amplify smaller fragments, okay, and then you have, say, you run 5-6 PCR reactions. So this is a DNA strand, okay. You run one reaction from here to here, then another here to here, and then here to here, here to here. Then you have smaller fragments like these. Then you put them in a same tube, and then you run a reaction. So what will happen when you ligate them at TM? These will have these regions that are overlapping. So they will renature with these regions. And then the PCR Taq polymerase will join these regions. Okay, so assembly PCR is usually used if you have larger genes and you want to clone them, you want to amplify them. We do use the assembly PCR and all that. Then you have a digital PCR. Okay, in a conventional PCR, you have a tube, and then you amplify it and blah blah blah. So in a digital PCR, what you do, you take the DNA, okay, and then you dilute it and put it in different valves in 96 well plate. Okay, size that the dilution should be size that only one type of DNA should be in one valve. So when you run the PCR, okay, some of the valves will amplify and some of the valves will not amplify. Okay, so then you'll be able to see if you probe it with the fluorescent label. You'll be able to see that. So from here, the reds are positive one and the gray ones are the negative ones. So you'll be able to run multiple hundreds and thousands of reactions in a single time. Another type of PCR is Sher-e PCR. I have a VR or someone, so I usually ask this question. So so what we do here in a suicide PCR, first, for example, we have an archaeological sample from which we want to identify this archaeological sample. But we know that there can be some other contaminant that is present in the archaeological sample. So first type of PCR we run with the primers targeted to the unwanted gene. For example, it has a contamination of micro or a virus or something. Okay. So when we amplify this, okay, then we heat it at 95 degrees centigrade to make it single-stranded DNA. Okay. Then we cool it, okay, and we use air dissociation enzyme with DNTPs. So the primers near to the complementary sites, okay, and DNA polymerase begins elongation. However, the non-targeted DNA remains single-stranded. Okay. So now what happens since we are at restriction enzymes? Once these non-wanted DNA are amplified, they are restriction digested by our restriction enzymes. So we're only left with non-targeted DNA. It means, okay, it remains single-stranded. Okay, then we inactivate the endonuclease. Okay, and then this we can since it's an enzyme, we can degrade it or destroy it at higher temperatures. Okay, then the template was left, we can use it for sequencing reaction, and we'll be able to know that what we need. Then another type of PCR is the reverse transcriptase PCR. So here, when you have a DNA and you use a DNA-dependent DNA polymerase like a Taq polymerase, so that's a normal PCR. But however, if you have an mRNA and you want to make a cDNA from it, then you use reverse transcriptase PCR, because the enzyme here you are using reverse transcriptase that is isolated from another way, or with a very synthetic or recombinant form of reverse transcriptase also. It's an RNA-dependent DNA polymerase that makes DNA from your mRNA. So again, you want to know more about this cDNA synthesis, you can go to the get it. So here you can see that You will add a oligo-DT and then you add a reverse transcriptase. So DNA strand and it's a DNA RNA hybrid. Okay, then it has an RNA H activity where the RNA is degraded in an DNA RNA hybrid with RNA H activity of reverse transcriptase. Okay, then there are three ways in which you can make another strand, like you have self priming here, or you can kind of do an extension, like you add GGGs here using thermal transferase and use the poly-C primer, poly-D primer. Okay, and then at the end, if you're using the self-annealing, then you use an SN nucleus that cuts the hairpin loops, and you'll have a double strand. This is reverse transcript PCR because we use a reverse transcriptase and by making a DNA from a messenger RNA. You have a semi-quantitative PCR. For example, if you want to know the expression of your genes, simply what you do, you take the total RNA and then you make a cDNA and then you use the PCR to amplify 200, 300, 400 base pair gene. Also, you'll amplify a housekeeping gene also, and then you will see the relative expression, that how much this band is, because the band of this gene of interest is in comparison to the housekeeping gene. It will minus it, okay, and then it will see the relative expression. So the intensity of bands tells us that the expression is more or the expression is less. This is semi-conjugative. So when I was doing my PhD, we were doing expression of phytosynthetase and phytodesaturate genes from bacteria. We did not have a real-time PCR, so I used this in quantitative PCR. So you can see the bands here. You can see here are of the PSY, and here the control we use as 26 levels of RNA. You can use gap data also. So then we can see the intensity of these bands related to these, and we can kind of. So these are the genes that are grown from black carrot, and we're able to reconstruct the whole terminal battery pathway for E. coli. So these are lipoprotein producing bacteria. I want to go in detail. Then you have methylation-specific PCR or MS-PCR. So here you treat the samples. You have say for the two samples of DNA, one is unmethylated and one is methylated. You treat them with bisulfite, okay, and then you run a PCR. Okay. So you'll be able to determine that these DNA are methylated or then you have a real-time PCR in which you'll be able to kind of determine the amplification curve in the real time. Okay, this is usually used for expression analysis. We do have a real-time in the lab and we run these experiments. So there are various dyes. What happens when DNA amplifies? Double-stranded DNA dye is bound like the Sybr binds to double-stranded DNA and gives a peak. So more double-stranded DNA is found, more will be the fluorescence. So in real time, you will know that if you have a more fluorescence, that means the gene is highly expressed. Or less fluorescence, that means gene is less expressed. There are many dyes like Sybr or green, or cyto family using this. So in a Sybr PCR, simply you can see that the dye fluoresces when bound to the DNA strands. So you have a single strand, the dye is not bound, but when you add primer, then you amplify it, then you form a double strand, then fluorescence is checked on the PCR layer. Then another type of dye is the TaqMan polymerase that we use. Okay, so it hybridizes. We use a probe in this TaqMan polymerase. In the Sybr, we do not use a probe. Simply the dye binds to it. So once you run the PCR reaction, okay, these probes, okay, they bind to this DNA, and when the DNA is amplified, they hydrolyzes. Okay. Or hydrolyzes, this gives a fluorescence. Okay. The R is called a reporter, it is called a quencher. When the reporter is bound with the quencher, it does not show the fluorescence. However, when the amplification takes place, more and more amplification takes place, this Q and R, they hydrolyze. Once they separate, these are is your report of these fluorescence and will be able to detect fluorescence. And more the fluorescence, more strands are synthesized, that means more. Then you have hairpin probe molecular like molecular beacons. Again, they have a quencher dye and a fluorescent dye. You run a PCR reaction, okay, they separate and then see the fluorescence. And fluorescence will tell you that it's accuracy more or it's accuracy less. Now, the application of in molecular diagnosis, you can have clinical microbiology or food microbiology, like some pathogens you want to see pathogens in the PCR. You can do gene expression of pathogens, like I talked about. It's a viral like COVID-19 currently going on. So if you run real time, so this is an RNA virus, so you have to run real time PCR. So reverse transcriptase PCR. Once you want to run the reverse transcriptase, then you run it in the real time PCR. Then you see the expression. If viral load is more, you have more fluorescence. Viral load is less, you have less fluorescence. So we can do a quantification of viral load. So some of the people cannot be symptomatic, they cannot have COVID-19, but though they have an infection. Okay? So maybe they are immune and the viral load is less. So that will be determined by quantification. You can kind of do simple nucleotide polymorphism, simply use a PCR, you amplify it, then you see a change. And sometimes these single nucleotide polymorphism can be lost by some genes. If they are mutated at a single base, they are highly makes highly lethal to the organism. You can use in clinical oncology to study expression of various genes. Okay, like some genes expression is increased, like you have a DHFR gene, the hydrofolate refractory gene, copy number increases in these cancers. So if you run a PCR reaction to see the copy number of the gene or the expression of the gene, you will know, okay, now the gene amplified, that means probably that this